Good day, Tahuro. I am Vanessa Mahad, your reporter today. Now we will discuss to you the classification of bacteria based on Dr. Bergy. First bacteria we'll discuss to you is morphology. Second, the staining reaction. Third is cultural characteristics, biochemical or physiological behavior, genetic analysis, or we'll know the animal inoculation. For last is immunology difference. Now we will discuss to you about the bacteria of Yersinia. Yersinia is a bacteria are typical small gram-negative roots. They can facultative anaerobics, meaning they can survive with or without oxygen. They have the characteristic fibular staining pattern appearing as a safety pin shape when stained with a certain taste like a gemma or right stain. The staining reaction Yersinia bacteria is a stain gram-negative. The cultural characteristic is a Yersinia bacteria typically grow well on routine laboratory media such as blood, agar, McConney agar, and nutrients agar. They appear as small, smooth, and sometimes translucent, colonized after overnight incubation at 37 Celsius. Now we will proceed to the biochemical or physiological behavior of Yersinia. Yersinia bacteria exhibit various biochemical and physiological behavior that are important for their pathogenicity and survival. Some key characteristics include the fermentation, second oxidation, catalyzed motility, hemolysis, biofilm information, and biflorance factor. Now, the genetic analysis, now we will know the animal inoculation Yersinia are the crucial for understanding the virulence mechanism, host interaction, and pathogen of these bacteria. By conducting genetic analysis, research can identify the virulence genes, regulatory elements, and other factors involved in Yersinia pathogenicity. Animal inoculation study often performed in mice or other animal models. Help research assess the virulence difference Yersinia stain, study of course infection, evaluate immune response, and test potential therapeutics, vaccines. This study provide a value science into the biology and pathogenicity of Yersinia species and contribute to effort to develop effective strategy for the prevention and treatment of Yersinia related to disease. The immunologic difference of Yersinia. Yersinia species exhibit immunological difference primarily related to their surface antigen, virulence factor, and ability to evade host immune response. Some key of immunological difference includes first includes is antigenic variation. Second is capsule production. Third is type 3 selection system. Fourth is liposaccharide or we know LPS. Third, biofilm information. This immunological difference contribute the ability of Yersinia species to cause resistant infection, evade host immune surveillance, and establish successful colonization with host tissue. Understand this difference crucial for developing effective vaccine and therapeutic against Yersinia-related disease. That's all. That's the bacteria of Yersinia. Now we'll proceed to other reporter.
This is Bacteria Cultural Reporter from Group 1, Rina Tandin. Today, we will discuss the morphology of Listeria. Listeria is a small gram-positive with a size of 0.5 to 4 micrometer in diameter and 0.5 to 2 micrometer in length. Staining reaction. The causative organism Listeria monocytogenes in a gram-positive bacillus that may resemble detroits on gram stain. Structural characteristics. It is rounded or sometimes crucial ends. They do not form spheres or capsules. Listeria are motile with perticious flagella at 20 to 25 degrees Celsius, but not are the motile, or less than noticeably motable at 37 degrees Celsius. Biochemical or psychological behavior. The biochemical profile of Listeria includes catalyzed positive, oxidate negative, fermentation of carbohydrates to acid but not to gas, and too many to mention. The genetic analysis of Listeria is confirmed only by isolation and identification of L monocytogenes. Specimens of choice can be tissue from animals with CNS involvements and aborted placentas and fetus. Immunologic differences of Listeria is L monocytogenes survive and multiply at low temperature, usually found in refrigerator.
Good morning, ma'am. My name is Juana Mamulama Samir from DSN1 Group 1. So now, let's talk about Shigella. Shigella is a genus of bacteria that is known for causing gastrointestinal illness. The morphology of Shigella bacteria includes being rod-shaped, bacillus, non-motile, and non-spore forming. They are also gram-negative, meaning they do not retain the crystal violet stain used in the gram method. These bacteria typically operate in pairs of chain and have a characteristic appearance when viewed under a microscope. Shigella bacteria can be identified through a specific cultural characteristic, such as producing colorless colonies on mangkoki agar, not fermenting lactose being non-motile, and non-fermenting lactose. These traits, along with biochemical tests, help differentiate Shigella for other bacteria.
Good day, everyone. I am Baitunggal Abdi, the morphology of Microbacterium. Microbacterium species can vary, but generally they are aerobic, rod-shaped bacteria. The weight of Microbacterium species range from 0.2 to 0.6, while the length can be between 1.0 to 10. Staining reaction. Staining reaction commonly used for Microbacterium species is the Jan-Nelson acid-fast stain. This staining technique is designed to stain bacterial cells containing long-chain fatty acids such as Microbacterium. Cultural characteristic. Microbacterium species, including Microbacterium tuberculosis, can vary depending on the medium used for cultivation. Biochemical and physiological behavior. Microbacterium species, including Microbacterium tuberculosis, exhibit specific biochemical and physiological behavior that contribute to the pathogenesis and survival. Genetic analysis and animal inoculation. It plays a crucial role in understanding the infection pathways and molecular mechanism of Microbacterium species, particularly in animal infection. This is immunologist differences. Immunologists are scientists who specialize in the study of immune system, its function and its interaction with pathogens, diseases, and overall health of an organism. There are different types of immunologists. Clinical immunologists are medical doctors who specialize in diagnosing and treating disorder of an immune system. Research immunologists focus on conducting scientific research to advance our understanding of an immune system. Veterinary immunologists studying immune system of animals. Molecular immunologists study of molecular mechanism underlying immune responses. Transplant immunologists specialize in the field of organ transplantation and immune responses associated with it.
Good day, my name is Norhema G. Ampaton. I'm going to talk about Listeria. Morphology. Listeria are short, gram-positive, rod-shaped bacteria. Staining reaction. They stain positive with the gram stain technique. Cultural characteristic. Listeria can grow at refrigeration temperature and can be found in various environments. Biochemical or physiological behavior. Listeria are facultative anaerobes, meaning they can grow in both the presence and absence of the oxygen. Genetic analysis. Molecular techniques such as PCR and sequencing are used for genetic analysis of Listeria strain. Animal enucleation. Animal models, particularly mice, are commonly used to study Listeria infection and virulence. Immunologic differences. Immunological study focus on identifying antigens and virulence factors of Listeria. Thank you.
Good day, ma'am. Hello, everyone. I am Joshua Melanjan Alberto, and I will discuss about Rhizobium. Rhizobia are rod-shaped bacteria with 0.8 um in diameter and 2 in length, often with flagellate. Its staining action is Rhizobia are gram-negative bacteria that can elicit the formation of specialized organs called root nodules on leguminous house plants. Slow-growing Rhizobia show an alkaline reaction in this medium, turning the dye blue. Fast-growing Rhizobia show an acid reaction, turning the medium yellow. Rhizobia grow poorly, if at all, on peptone glucose agar and cause little change in the pH when incubated at 23 to 25 degrees Celsius. Biochemical or psychologic behavior. Rhizobia are gram-negative bacteria which inhabit the root nodules of most leguminous crops. They are soil bacteria that fix nitrogen after becoming established inside root nodules of legumes. Genetic analysis. In the last few years, there has been an increasing interest in the genetics and molecular biology of Rhizobium, the bacterium that is responsible for inducing nitrogen, fixing nodules on the roots of leguminous plants. The recent studies implicating indigenous plasmids as important determinants of some symbiotic functions, and finally the use of Rhizobium mutants as aids in the biochemical analysis of the symbiosis.